Good morning! Welcome to SR Life Science. Subscribe my channel. Let me know your opinion in comment box. And share with friends. The outcome of any biological experiment is data.
Previously, biologists analyzed data without the help of software, computational tools, and statistical tests.
With the advent of instruments like DNA sequencers,
Microscopes, and other imaging systems, biologists can no longer deal with the data using their notebooks and Excel sheets.
instead, they need computational and statistical tools to handle data.
Large volumes of data often require quantitative analyses to interpret and generate biological meaning.
Performing such analyses require good working knowledge of computational
And statistical concepts.
for example, machine learning technologies, regression, variance, and correlation. Mathematical and statistical concepts can only aid
Biologists to interpret their data.
and are not a replacement for asking the right
Regression,
questions
variance,
and the biological acumen. Bioinformatics uses computational, mathematical,
statistical and engineering approaches.
and analyzing biological information for solving biological
problems.
and the biological acumen.
Thus, bioinformatics deals with storage, retrieval, analysis, and interpretation of biological data.
Using computer-based software and tools.
It is interchangeably used with other terms such as computational biology, quantitative biology, and biostatistics.
Depending on the dominating disciplinary components. It must be noted, however, that use of these definitions
Vary among experts and practitioners and has changed with time.
I love exploring these mountains.
Me too. And it's like we discussed in class.
Right, FASTQ and FASTA are just the start.
Like homology search and sequence alignment. Then there's phylogenetics and gene prediction.
Don't forget RNA structure and protein structure, and regulatory region analysis.
We can use tools like BLAST or CLUSTAL
There's also MAFT, muscle.
for gene predicts and gene prediction.
We have GlimmerNChem and GenScan, and Fire2 or JPRED for protein structure.
Bioinformatics tools use various statistical and computational algorithms and approaches like SPSS, SAS, and R.
And don't forget Microsoft Excel. At the final stages of the biological data analysis,
Gene and protein level findings are linked to certain
which can be made
functions.
by using biological tests like loss or gain of function assays.
Additionally, inferring affected networks and pathways by using computational tools.
One can assign functional significance to a gene and its protein product.
That is very true.
A biological database is a repository that contains an organized, structured, and searchable collection of biological data. In other words, it is a library of biological information.
A biological database links all the relevant data to their original creators or scribes the underlying data.
The information is collected through experiments and computational approaches. For example, a database of human genes contains both the actual nucleotide sequence of all the genes and their characteristics.
A biological database may store multiple types of
Right,
information.
and sequence of a gene.
Mutation in a given gene specific to a disease. And the frequency of SNP in various population.
So what makes a good biological database then?
Well, it should be easy to access.
And have a user-friendly interface.
It also needs support staff to answer any queries from its users. There are two main types of databases, relational and non-relational.
Aren't they managed by a specific system?
Yes, it is called a database management system, or that is used to manipulate, retrieve, and manage data. Did you know that structured query language
Or SQL is the standard API
for a
for
relational database? A non-relational or NoSQL database
does not follow the order of a relational database.
and is used for large sets of distributed and unstructured data. The database can be made by a single group's
Collecting information from a variety of public resources.
or by multiple teams of researchers who can add data to a single repository. Some commonly used biological databases are GenBank,
PDB, UniProt, PubMed, KEGG, and OMIM.
Some commonly used biological databases
There are organism-specific, disease-specific, and secondary databases, and are used routinely by biologists.
Some commonly used biization is an essential aspect of bioinformatics.
It involves the application of graphics and data representation and incorporates sequences, genomes. Genome is the complete set of DNA of an organism, including its genes.
And the intergenic regions.
Genomics is a field of science that deals with the structure,
Function, evolution, mapping, and modification of genomes.
Genome informatics is the application of bioinformatics tools
to process the outputs of genome-wide assays and technology of genomes.
Genome information provides data on DNA and RNA nucleotide
and
sequence.
changes in gene expressions,
We can also see profile genomes.
along with DNA and expression.
We can also The term high throughput relates to that produces a significant amount of data.
The amount of data generated by genome sequencing is substantial. For example, some large genome centers in the world
Like MIT and Harvard?
produce more than 24 terabytes of genome data per day.
Furthermore, the magnitude of such data generated is increasing continuously, day by day. Although there are not many large genome institutes, in fact, it is postulated that the computing resources required to handle genome data will exceed those involved in processing Twitter and YouTube data. As the amount of data is significant, genome data need the power of information technology and
for
statistics.
gaining an understanding of underlying complexity, patterns, and meaning. An initiative to sequence the complete nucleotide content in human cells started in early 1990s. This initiative is known as the Human Genome Project. The method proposed by Fred Sanger was used both the publicly funded initiative headed by the NHGRI of the National Institutes of Health USA and the private company Celera Genomics. The publicly funded initiative sequenced human DNA
cloned into BASE, and Celera Genomics sequenced randomly cut human DNA, a technique called whole genome shotgun sequencing method.
Did you know that the first complete draft human genome was
Published in 2001,
Yes, and most sequenced part of the genome is in the euchromatic region.
which is found in the chromosomes, right?
And with minimal the heterochromatic areas mainly consist of repetitive elements and are primarily located in the centromeric regions.
They are difficult to assay with the current sequencing technology.
Additionally, due to the repetitive nature of DNA, and are
To be
primarily
assembled to a single location, in the chromosome, unambiguously.
One of the challenges of bioinformatics is
Different formats of the resulting
the
data.
bioinformatics community adopts a standard data format. For example, most DNA sequencing data are presented in
FASTQ format.
text-based format that stores sequence information. For example, most DNA's quality score use a single ASCII character.
The FASTQ sequence uses the FASTA format.
FASTA format is a text-based format for representing sequence information in single-letter codes. The first line usually starts with a greater than symbol. It holds a description of the sequence. Often it's a unique accession number or gene name. A FASTQ file uses typically 4 lines per sequence. The first line begins with a @ character, followed by sequence description.
line 2 has the raw sequence letters. Line 3 begins with a plus character. And line 4 shows the quality values. The quality values in line 4 contain the same number of symbols as letters in the sequence presented in line 2. Both line 1 and line 3 may include optional sequence identifiers. The characters and N represent the lowest and the highest quality respectively.
Genome informatics tools came up in parallel with the development of sequencing technology
To cater the analysis of resulting data.
Genome informatics tools, the high-throughput sequencing instruments produce sequence reads, either short or long, depending on the device.
Wow, that is fascinating.
Resulting sequenced reads need to be either assembled into a genome or aligned to a reference genome, in the case of resequencing. Broadly, there are 2 possible analytical workflows, one based on genome,
And second, based on the de novo assembly of reference genome sequence.
In both the scenarios, the sequencing data is pre-processed and checked for quality.
The de novo assembly-based workflow?
It involves assembling a genome based on the overlaps and insert size. The assembly undergoes further annotation and processing, quantification of gene expression, and identification of splice variants, novel isoforms, and fused transcripts.
After a genome is assembled, it is analyzed for predicting and identifying locations of coding parts of genes. Repeats are a significant component of any eukaryotic genome. They provide diversity and could play a significant role in their evolution. Genome annotation is the process of identification and classification of all the features in a genome. Repeats in a genome are classified into 3 major categories: transposons, satellites, and low-complexity regions. Gene prediction involves identification of all the coding sequence in a genome. Tools used for gene prediction may follow either ab initio.
The similarity-based approach uses sequence similarity for identifying coding regions.
The last approach to identify genes both strategies and integrate the gene prediction.
After gene prediction, the gene products, and their associated functions,
The last approach to be integrated and represented without ambiguity.
the gene ontology aims to describe gene products.
Regarding their associated biological processes and molecular functions.
Phylogenetic analysinary relationship of an organism with others.
It can be expressed as a diagram called cladogram or phylogenetic tree.
There are different methods for calculating evolutionary distances.
These are used by tools for phylogenetic analysis like phylip and. Thank you for watching. See you in the next one. Stay tuned.